So our first speaker for this session will be Lucile, Massenet Regad, and she will talk about intercellular communication using the ICELLNET framework. I think it's going to should make interesting bridges with this morning's keynote from Sarah Teichmann. So Lucile, floor is yours. Thanks.
So it's okay, you see my screen?
Yes, fine.
So hello everyone, I'm very happy to be here, and I would like to thank the organizer for letting me the opportunity to speak about my work on intercellular communication using ICELLNET. So as you may know, cells communicate with each other by secreting soluble molecule and also cell surface molecule that will be sensed by the other cell, the target cells, with their specific receptors. And cell communication is involved in many cellular processes, such as cell development, tissue homeostasis, and also in disease, for example, in cancer with immune and cancer cell interactions, but also many other contexts. So in the lab, we are aiming at deciphering the complexity of cell communication networks in health and disease. And for that, we have several focus. First, study the diversity of cell types and its plasticity, and also to see how cells integrate different signals, if there is synergic effect or not. And all of this aim at understanding better the cellular networks and how it works together in a more complex systems such as cancer. So to study cell communication, most of the methods that exist are based on transcriptomic data, and most of them are based also on single cell data sets, where the cellular profiles are determined, analyzed, and then they are focusing on interaction between ligand and receptor pairs that are knowledge-based. And then, depending on the method, you have different interpretation and visualization of the methods. So as we heard this morning, there is CellPhoneDB, which is the most used method to deal with this intercellular communication. You have also NicheNet, which is a bit different, and others, and they all have some advantages. And I think that it's useful to use several to have different visualization and to help in the interpretation, which is the most challenging thing actually. So today I will present you ICELLNET. So ICELLNET, the idea was to not deal with single cell data, but also use the other type of data that are available in the literature. So for that, we are focusing on also cell-sorted population in bulk RNAseq or microarray data. And this is also possible to apply with single cell RNAseq. So let's say that we have a central cell that we have the transcriptomic profile, and we want to decipher the communication. We offer the possibility to do it using the Human Primary Cell Atlas, which is a published data set across 31 cell types. And this is kind of a reference cell type that we can compute the communication with. So in ICELLNET, we also integrate a knowledge ligand-receptor database that we manually curated, including all the interactions that are experimentally validated in human system. It's the one of the database that integrate multiple subunits of ligand and receptor, which is really important for immunology. And the interactions are classified into different molecular families and subfamilies. So today we have more than 700 interactions in this database that are classified in 7 families of molecules, but it's a continuously growing process, the most recent version is always on the GitHub page. So this is how the database looks like. So it's really easy to understand. We have the genes coding for the ligand subunits and receptor subunits, the classification into family and subfamily of molecules, and the references. So here is the repartition of interaction according to the family. And for cytokines, we really go deeper into subfamilies of cytokines, and we classify them according to the structure of the receptor. So then to study communication, we compute a communication score. They're based also on ligand and receptor expression. So let's say that we have the central cell that we want to compute the communication with a partner cell that can be any of that. So let's say in the human cell atlas, but also from other data. And what we do is we can compute an outward communication score considering the ligand of the central cells and the receptor of the partner cells. And we will multiply the expression of the ligand and receptor for each interactions. And so if the two are here, it's not zero, otherwise there is a zero for this interaction, and we sum up for all the interactions. So this is a score, and then we have multiple level of visualization. The first one is a quantitative view where the network is shown and you have an arrow according to the score. You can also have a qualitative view where we can see the distribution of the score and the relative proportion of each family of molecules that is contributing to the score, and another level of visualization where we focus on specific interactions, for example, to see the most contributing interactions to the communication or the most different among conditions. And all the representation will be soon available to the community. So this is for the tool, and I will show you very quickly two applications that we did with ICELLNET. So this first one is focused on an in vitro system where we have a DC that we activate in vitro with LPS, and we block the TNF loop or the IL-10 loop. And so we have these 4 experimental conditions where we generated microarray data, and we looked at the communication with the other cell type of the primary cell atlas. And what we see is that we have an increase of communication when blocking the IL-10 loop compared to the other conditions. And then with ICELLNET, we can have the different specific interactions that are increased and showing an increase in communication with other DCs, neutrophiles, and keratinocytes. We then further validate by looking at the protein level of DC, and we see that for each condition, we really have an increase of some protein secretion when blocking the IL-10 loop. So IL-6, OSM, IL-23, and IL-12P7T were increased when blocking the IL-10 loop for LPS activated DC. To go further in the validation, we then put some target cells in culture with the DC supernatant of each condition, and we measured some activation markers of the keratinocytes and PDCs. And what we see is that by blocking the IL-10 loop, we really have an increase of communicate activation in both systems. And to go further, we blocked the 4 most known ligands that are known to activate keratinocytes or PDCs, so they are here and here. And what we see is that blocking TNF is sufficient to inhibit the activation. So this is about this in vitro model. And now I will quickly go to another application to single cell data, this time about the tumor microenvironments. So dendritic cells are known to participate to the anti-tumor immune response, and they are also essential for immunotherapy efficacy. And some studies have already suggested that factors derived from the tumor microenvironment can induce tolerogenic disease. So we focused on that because in the lab, we showed that 2 states of human activated DC can arise from different stimuli activation, and they can be discriminated with PD-L1 and ICOS ligands. And we show that the PD-L1 ICOS ligand, PD-L1 high, ICOS ligand low show a secretory DC phenotype. So these DC express a lot of cytokines, but they are not really efficient to activate T cells. And at the inversary, we have the helper DC, which are PD-L1 low and ICOS ligand high, that are not secreting a lot of cytokines, but are very efficient to induce TH cytokines after co-culture. And what we see is that in a single-cell RNAseq in head and neck cancer, we are able to see this secreted DC. secreting DC. So we are here the different population of the single cell data sets. So the lymphoid and myeloid compartment, also cancer cells that are here. And for the clusters of dendritic cells, we have the conventional dendritic cells, which is the cluster 7, and the mature dendritic cell, which is the cluster 20. And what we see is that they exhibit a tumor secretory signature here. So what we wanted was to decipher the communication and compare between these two clusters of DC and the other component of the tumor microenvironment. So to apply ICELLNET, the only thing that is changing in single cell is that we do an averaging expression for each cluster, which make the profile, the meaning profile for each population. Then we filter to keep the genes that are expressed by at least 10% of the cluster, let's say, and we apply ICELLNET, so we have the communication scores and the different visualization. And what we see first is that we have an increase of communication from secretory DC with the other component of the tumor microenvironment compared to the conventional dendritic cells. And this is observed with all the cell types and the two sense of the communication, let's say the inward and outward communication. Then what we saw looking at specific interactions is that the secretory DC acquired the migratory, co-stimulatory, and inhibitory functions according to ICELLNET prediction. So here are all the interactions that were really highly expressed by this, the 20 clusters, but not really present by the 7 ones. So in conclusion, ICELLNET framework is available on GitHub. You can really, I encourage you to use it, and really don't hesitate to contact us to implement other things in the tool. It's very adaptable and easy to use, and ICELLNET can be applied on several types of data, so single cell, but also microarray and bulk RNAseq data. It allows the network reconstruction from data of one population if we use the primary cell atlas as partner cells, but also several different populations from the same sample. Let's say if we sort a different sample of the population from the same sample or with a different cluster of single cell RNA-seq. And several interactions can be found in the in vitro LPS activated DC model were experimentally validated. So we can now use it in many topics to, for example, help in deciphering the DC communication with the tumor microenvironment in head and neck cancer. So thank you for listening to this presentation. And now I would like to acknowledge all the team that has tested the tool, and also the person that participated to this work, so in particular Florian, and also Caroline and Philemon that did all the single cell study, and Irit and Antonio that take care about the DC in vitro model a bit. Thank you very much.
Thanks very much, Lucile, and thanks for your timely, quick talk. We actually have time for a lot of questions. Let me open. So to ask questions in the attendees, you can either just type it in the question and answer section, or raise your hand, and hopefully I or someone will see it. Attends, Q&A, clap, clap. Yes, clap, clap. Up. So Stefano Colella has a question. Can somebody give microphone rights to Stefano?
Yes. It's okay. You can talk, Stefano.
He has to unmute himself, I guess.
Oui, c'était une erreur, je pense. Parce que
okay.
Je ne sais pas qu'est-ce que j'ai cliqué, désolé.
It's fine.
Mais merci, c'est bon topo, bravo.
Oh, you mean you did not have a question? Okay, so we have a question from Vu-Mantien. Vu, I think you can talk now. You have the microphone raised.
Hi, can you hear me now?
Yes.
Yeah, sorry. Thank you very much for the talk. It was very interesting. I was wondering, because you mentioned other softwares such as cell phone DB, what are the advantages or the drawbacks as compared to ICELLNet?
So I think that the first thing that cell phone DB do is that it's a bigger, actually it's a bigger database that is used. And also there is a statistical framework in cell phone DB that you don't have in ICELLNet. But in ICELLNet, you have also many like graphical interpretation. We really focused on a graphical representation that you don't have on all these tools, like all at the same time. And the idea is really to have layers of interpretation to go to a global score, but also on family of molecules and then specific interactions. This is the big focus on of this method.
Okay, thank you. And is there any, are you planning to integrate the localization of the cells in order to?
So this is part of the perspective, but for now we didn't focus on that. So this is a challenge to integrate other type of data, but for the moment it's more manually done, let's say.
We have a question in the Q&A thing. Clémence Reda is asking, so thanks for your talk. What is the score used by ICELLNet? In particular, it seems asymmetrical, right?
Yes, so I forgot to mention that. So actually the score is really just the multiplication of the ligand expression and the receptor expression in one direction. So let's say we want to see the outward communication score, we will consider the ligand from the central cell and the receptor of the partner cells. But if we want to look at the communication in the other direction, so from the partner cells to the central cells, then the score will be computed differently. It will be the ligands expressed by the target cells and the receptors by the central cells. So it's really depending on the direction of the communication you want to see, the score is computed either with ligand or receptor of each cell type. I don't know if it's more clear. I hope.
C'est bon, Clémence? Yes. So we have time for one more question. Wait. Attends. Thanks a lot. It is clearer now. Clémence is typing. We have time for one more question if somebody wants. Otherwise, we will move to the next talk. One last question. Une fois, deux fois, trois fois. Okay, well, thanks again, Lucile, for a nice talk.
Thank you.
Attends, alors.